Well, thank you for the introduction and thank you for the invitation. It's actually over a year now that I've been presenting in front of the real-life audience, and I have to tell you, I'm a little nervous. So I have to go back to the playbook of presenting that tells you in a situation like this, you have to picture your audience naked. That's better. I think we're good. So let's start, shall we? Standard semen parameters like count, motility, and morphology of sperms are more laboratory findings than an actual diagnosis. Among those thank you for taking a picture. Among those diagnoses, urogenital tract inflammation and infections are thought to make up to 15% of the underlying causes. And these causes are known to damper or disturb such parameters like motility, morphology, and also sperm count, but also sperm function. But keep in mind not to confuse inflammation with infection. They are not equal. As in 50% of cases, the microbiology workup remains negative. So how do we diagnose inflammation? Inflammation is diagnosed by leukocyte count exceeding a number of 1 million cells per milliliter. And how do we do this? By peroxidase. By only counting peroxidase cells, positive cells, which isn't that easy. If you look at the picture here, you have to say clearly, this one, it's a leukocyte. And it's not degranulated. But if you look at this leukocyte here, it's an intermediate state. If you ask my technicians, 50% will say that granulocyte degranulated, we don't count it. The other half will say, no, it's still positive, we count it. This one here, this leukocyte here, clearly degranulated. It's shoot its ammunition off, and it's not counted. So this tells you the bias of this method. First, it's strongly dependent on the eye of the beholder. Secondly, it doesn't even detect all leukocytes. For example, macrophages do not even express peroxidase. So you won't count any of the macrophages, which are a big player in the immune system with this method. And last but not least, third, you only detect inactivated cells, not the activated. It's like saying you up here in the front, you got a loaded gun, it's all locked and loaded, yeah, you're a threat. Fair enough. But Dorian over there, he got a gun and fired it all up, unloaded, might have killed a few people, but we don't know if he has any further guns. He's good, no threat. So what I'm trying to get across is that with this method, you cannot detect inflammation within the seminal plasma in an accurate way. A more accurate way would be using flow cytometry, like the rest of medicine. And we conducted a study using CD18 as a pan-leukocyte marker. Other groups use CD45, that's fine too. And we could detect a strong correlation between flow cytometry and peroxidase method. But what we could see that in samples where the peroxidase was negative, below 1 million per mil, we could detect 30% of samples that were positive, exceeding 1 million leukocytes per milliliter if you use flow cytometry. Furthermore, flow cytometry allows you to distinguish several pictures within the immune reaction. In this case, for example, we only detected neutrophils in a sample that had a peroxidase of over 1 million leukocytes per mil. Only neutrophils detected here by CD66. But in other samples, you find this pattern. You find 50% macrophages and 50% neutrophils. Clearly, those two immune reactions are different from each other, have different functions, and might harm sperms in a different way. But with the peroxidase method, you will not capture these different immune reactions. Yeah? It's like one is heavy metal, one is Beethoven's symphony, one is gentle to your ear, the other one scares you. So the difference between the immune reactions are not covered clearly with the standard method proposed by the WHO. And if you look at the distribution of macrophages within the ejaculate, you will find that in around about a quarter of samples, you have 50% macrophages within the leukocyte fractions, and in 35% of samples, you would find at least 30% macrophages. Another method to detect inflammation are biochemical markers, such as interleukin 6 or 8, or reactive oxygen species resulting in oxidative stress. And if you look at the source of cytokine production, you will see that rather macrophages are producing interleukins than neutrophils. Here shown by TNF-alpha, IL-1, and IL-6. And if you look at the cytokine network, it functions like a key and a lock, whereas the key is interleukin 6 in this example, and the lock, its respective receptor, the interleukin 6 receptor. And we checked the expression of the interleukin 6 receptor on sperm, and we could find its expression on the midpiece of the sperm. And the receptor expression was highly dependent on the seminal plasma IL-6, and it had an inverse correlation to sperm motility, which is plausible. We then conducted a different set of experiments incubating sperms with IL-6, and you could see a slight but significant decrease of motility when incubating sperms with interleukin 6. Another agent in the inflammatory orchestra is reactive oxygen species resulting in oxidative stress. And those oxidative stress is not only present in infection and inflammation, also in smokers and in varicocele patients. And what you can see, there is a correlation between the interleukin 6 expression and the oxidative stress. It's not very robust, it's only 0.2, but it's significant. Why is the correlation so weak? Both are inflammatory reactions. Shouldn't they be a stronger association? No, because you will find entities where you have a high oxidative stress, like in the varicocele. In varicocele patients, you have high oxidative stress that is significantly higher than in patients without a varicocele but with inflammation. In both groups, you had a significant higher concentration of reactive oxygen species. If you look at IL-6, patients with a varicocele are less likely to have high IL-6 expression in the seminal plasma. There are some that have, on top of their varicocele, strong IL-6 in the seminal plasma. But most of the varicocele patients do not have high interleukin 6 seminal plasma, compared to, of course, patients with inflammation but without a varicocele. Nevertheless, if you look at the control group in both varicocele as well as inflammation without any varicocele, you will see significant higher IL-6 seminal plasma concentration. Also, what you can see in both groups, varicocele and inflammation without a varicocele, an increase of DNA fragmentation. That is comparable in both groups, the varicocele and inflammation without a varicocele. Both very bad fragmentation of the DNA compared, of course, to the control group that had no varicocele, no signs of inflammation. And like other groups, the Aitken group and also Agarwal, we could support their data that has already been published on this topic, showing that the higher the oxidative stress is in the semen, the lesser the sperm count is, the concentration, the motility, and the morphology. If you look at chronic inflammation, oxidative stress, and cytokines like interleukin 6 are involved in the generation of a chronic inflammation. Especially IL-6 drives regulatory T cells towards a Th17 phenotype. Those are T cells producing interleukin-17, resulting in a chronic inflammation and later on in autoimmunity. And Th17 cells also express the chemokine CCL20. And now you ask, so what? There has been a paper in 2013 showing that the CCL20 and its respective receptor on sperm, CCR6, are involved of the chemotaxis of the sperm to the oocyte. So we investigated this in the human system, and we could show that follicular cells indeed express high amounts of CCL20. So we then looked for the respective receptor on sperms, which is CCR6. And we could see CCR6, or we could detect CCR6 on sperms, and the sperm, or its expression, was on the tail of the sperm. In contrast to the IL-6 receptor that was on the midpiece, the CCR6 is expressed on the tail. And of course, we found various amounts of CCL20 within the seminal plasma, and interestingly, the amount of CCL20 correlated strongly, of course, with interleukin-17, but also with interleukin-6, meaning that in inflammatory status, CCL20 binds to the receptor of the sperm, CCR6, and then the sperms are more or less blind. That was our hypothesis that we tested. And as you can see here, this is chemotactic experiments that we performed with sperms, and we had capillaries filled with medium, and you see a random migration of sperm towards the medium, an irrelevant chemokine that we use, CCL21. You see the same migration pattern, and all of a sudden, if you use CCL20 within the capillary system, you see a strong migration of the sperms towards the CCL20 capillary. You can do that with human follicular fluid, same picture, but if you block the CCL20 within the follicular fluid, or if you use sperms from male with an excessive CCL20 concentration, this effect abolishes. So that proved our hypothesis that in males that have strong inflammation, strong CCL20 expression, their sperms are less likely to make it all the way to the egg. Of course, there can be always a lucky shot, but it is reduced in males, sperm males, of males with the inflammatory condition. And this is the last slide. This is where we took the sperms from an infertile man with high CCL20, and you see the reduction in migration to the CCL20. So in conclusion, I would like to say that inflammatory parameters are helpful for diagnosis. Although the techniques that are recommended by the WHO do not cover all aspects of an immunological reaction that is important in male subfertility. Cytokines and oxidative stress are valuable diagnostic tools, especially in patients with varicocele. And helpful tools are necessary from an andrology side in selecting andrological therapies, and of course the ART method, if you do IVF or ICSI. With this, I would like to close my talk. This is our center, where I'm the director of the andrology side. Professor Zenger, my colleague, she's the director of the reproductive female part, and this is my email address. If you have any questions, just drop me an email. I think we're through. Oh. Shh. Thank you for your attention.
Thank you, Pierre. Yes.